Thank you very much for the introduction, and even more so for the opportunity to present to you what we are thinking in the SGG, the Strategic Governing Group Oncology, what we can do in order to contribute to the microbiome field, where we see the opportunities, but also, of course, the challenges. So this presentation basically is shared by myself, and as you know, I'm heading up early development strategy and innovation in Novartis, and this is my co-chair of the SGG is Holger Hess-Stump. He's here in the audience, and he helped me also to put the presentation together. And on behalf of the SGG Oncology, I will lead you through a couple of thoughts, so to say, we have in the field of microbiome. So we kind of titled it basically Microbiomes, a Forgotten Organ. And what we know by now is basically that the gut microbiome is definitely a potent modulator of the immune system. This has been shown in many diseases, this has been shown in many cases, and it has also been shown in the field of oncology, and I come to that especially in the next one or two slides. On the other hand, basically what we have seen that by Tom Gajewski and Lawrence Stitvogel, they have really demonstrated that the gut microbiota could really modulate the efficacy and of the immune checkpoint blockade therapies. And this is something which is, of course, very much the trend of the time. So every immuno-oncology kind of compound is looked in how can we increase the response rate. You can go to PD-1, you can go to PD-L1 inhibitors, you can go to combination therapies, you can do whatever you want. The response rate, so to say, is around 20 to 30% of those patients. And this is also the patients who kind of will be on the tail of this kind of clinical trials and have a kind of really a long-term response to it. The question is, can we increase the response rate? Can we increase the patients who will be on this tail, who will have a long-term benefit from immune blockade, and not only basically a kind of a short-term benefit as we have seen it in the area of targeted therapies before. So you get a good response, but it's not durable. On the other hand, you could also demonstrate basically that the microbiome basically could predict, so to say, side effects. So which patient is really suffering side effects in a safety perspective, so to say, from your treatment. And also this helps you in a clinical trial, number one, to prevent it, and or to early intervene in order to make the side effects, so to say, not so impaired, so to say, for the patient's life and well-being. What you know here, basically, and what we have seen is, and this is data mainly from Lawrence Sitvogel's lab, is two things. Number one, if you look at stool samples pre and post-treatment, and that has been clustered A, B, C, and D, and this is a kind of various species, of course, in between, what you can see after ipilimumab treatment, the clusters shift. So that tells you basically that not only the microbiome has an effect how the treatment will influence the efficacy of the therapy, it also basically the treatment has an influence on the microbiome. And this relationship is something we need to figure out much better. So can we really work on that? Can we really reconstitute the microbiome in order to understand what treatment to give, and also in a longitudinal way? On the other hand, and this is also clearly came out of this kind of paper, and this is roughly 2 years old now, that certain clusters, A, B, or C, have a very different response rate, so to say, to ipilimumab. And that also tells you that it has a predictive power. The microbiome can really tell you which patient should be treated, which patient should not be treated. And that is something basically which is a huge potential for pharmaceutical industry, and also basically for patient treatment on a daily basis, which we want to explore further. But we also know what we don't know. And many of those things have been discussed, so to say, in the morning session so far. And there's a lot of things we don't know. And as it just has been said by the panel, I mean, the microbiome is a little bit hyped at the moment. So everyone talks about it, everyone wants to work on it, everyone sees the potential. But of course, we also have the challenges we have to face with this kind of new area of research, so to say, at least for the medical part. And the question is, what is a normal microbiome? I mean, it has been discussed many, many times. Maybe normal is not the right word, and healthy may be the better one. Maybe healthy doesn't exist either. And what's a healthy patient? Maybe it doesn't exist, it's just underdiagnosed. And those things is something which we have to evaluate, so to say, in that field. So what is the baseline? And what would we conceive, so to say, as a baseline, so to say, to look on differences, on the delta for disease and or treatment. The other thing is basically, how is the microbiome influenced by the diet? Who takes probiotics in the morning, yogurt, pills, whatever, may have very different microbiome than someone who doesn't. Smokers, different than non-smokers. And all of those things, lifestyle changes, vaccinations, and all of those things need to be taken into account because they certainly have an influence on the microbiome. Without knowing that, it's really hard to find biomarkers for it. And I mean, we have learned the same lesson, so to say, if you want, also in the area of the genomic medicine, where we identify mutational pattern in order to understand which patient respond. We still don't know, basically, take a melanoma patient and you can go to BRAF V600E, KDNR, and whatever you want. And that alone doesn't tell you, is this a patient, is this a responder to a BRAF inhibitor or not? And the same will be true in the microbiome, basically, if you take it to the immuno-oncology field and others. Then the question is pre-analytical standards. I mean, we have discussed that as well. How do we collect? When do we collect? How do we ship? And that's not a minor problem. And if you do from a pharmaceutical industry, global clinical trials, we're talking about 200 centers. We're talking about 300 centers across the world. And the standards are not the same. You go from America to Europe to Asia to other parts of the world. And you need to be clear that the pre-analytical standards, basically, because in a global clinical trial, we have to be honest, it's uncontrollable. We can't control for that. So the standards need to be very, very kind of, let's say, secure that we know what is going to be shipped to us and what we're really analyzing. Is it really referred to the patient, or has it been influenced by the shipment, by the storage, freezing, non-freezing, cooling, non-cooling? All of those things is something we need to understand much, much better from a pharmaceutical point in order to drive treatment decisions. And of course, also it has been discussed what to analyze: bacteria, viruses, yeast, fungi, everything. And how do we do it? 16S, whole genome. All of those questions are open. And I mean, that's the opportunity we have at the moment, and that's the opportunity we should grasp as an industry and as a private public partnership together. And last but not least, of course, we also have to think about how do we analyze it. So bioinformatics, what cluster A, B, C? What is cluster A, B, C? Is it sufficient? Do we need DEF? And how far do we need to go? And those things, basically, from an analytical point, are largely unsolved. So where do we see the potential in the SGGs, and especially in the oncology? Certainly on 3 to 4 areas. Definitely the microbiome as a therapeutic target. So can we really influence the microbiome to prevent diseases? Can we reconstitute it? Fecal transfers, other functional food, so to say. Is it something we have to give the patient in order to understand, so to say, that this patient changes the microbiome? With the microbiome, we prevent the disease of occurring, and or we prevent the disease of going worse. We can stabilize the disease. The other part is basically microbiome, and I talked a little bit about it as a biomarker. A biomarker in a way of, is it a predictor of response? Is it a prognostic tool? And is it, how good is it as a predictor in a way? Because if you talk about biomarkers, and this is something we also need to understand very well here, the question is not, can we measure it? The question is, how do we interpret it? Because at the potential, we withholding a patient from a treatment if we do the wrong thing with a biomarker. We may withhold the patient from an effective treatment if we don't really know how to interpret what we measure. And that's something which needs to be very clear when we're talking about microbiome, and the same is basically clear when we're talking about targeted therapy in the last 5 to 10 years, so to say. The other part is the monitoring tool. You have seen by the paper from Lauren Sitvogel that during therapy, the microbiome shifts, the microbiome changes. What does it tell us? For those patients where it shifts, is that the responder or is that the non-responder? For those ones where the microbiome remains stable, is that the patient who has the long-term response or has it been a shorter-term response? So this monitoring tool is the easiest thing you can do. It's non-invasive. Every patient will love it. There is no needle, there is no scan, there is no biopsy, there is nothing like that. So, and that's something where we need to understand as an IMI, as an industry consortium, so to say, the potential of it. And of course, basically, we need large data sets to drive basically a future drug design. What I mean with that is basically, if we know that ipilimumab influences the microbiome in a certain way, can we design the drugs differently in order to have a differentiated effect on the microbiome. And that's something basically which may be possible by adding some side chains to the drug. It may be formulation issues. It may all be those things which we don't know yet, but certainly there is a potential for that. So what's our hypothesis for that, and what we are currently looking into is really we want to look in a robust microbial composition signatures and the functional pathways. And that is really in order to understand how does it relate or not to an immune, or maybe even also to a targeted therapy or combination therapy. I mean, the next wave of oncology treatment is certainly combination of targeted therapy versus immune treatment sequences. We start with an immuno, we follow with a targeted, or we do the other way around. Those things are very interesting from a correlation with the microbiome and the influence of the microbiome to the response of the patient. Pre-treatment signatures as predictive biomarkers of response is certainly something which is extremely interesting for us to save patients on clinical trials, and of course also increase the success rate of clinical trials by having the right patient on the trial and treated, and saving the patients basically from non-response if we can. On treatment samples, as I said already, what does it mean, the microbiome change or not during treatment? How does that relate at the end of the day to an outcome to the patient or not? Can we design basically bacteria? Can we design functional food? Can we design something which helps the patient to be responsive to a drug longer, meaning the microbiome doesn't change? The microbiome stays the same, and that basically means the patient is still responding. And those are the data we do need in order to understand the potential of that tool. And of course also functional pathway analysis, so short-chain fatty acids, you have heard it in the morning, have an influence on the immune system. And we know already that they are associated with increased Tregs, and that basically is a consequence of potentially less responsive patients for those ones where this happens. So can we influence that again by diet and or by other mechanisms? The other part is basically what we said, okay, how can we get there, and how can we really make a difference, and what's the pathway to win? And here's a couple of vignettes and nothing more. Don't take that as a comprehensive statement. This is still in development in the SGG, just to be very clear here. So definitely analyze the microbiota in clinical trials, meaning we thinking of kind of collecting in the normal clinical trials the stool samples in order to analyze them and to kind of correlate it with the response rate. And this can be done across industry. This can also be done pre-competitively. That's not too difficult to do. Can we augment the baseline microbiota in order to enhance the response rate or the duration of the response? Is that something we were looking into? Drug development programs, I touched on that already. And we believe, and I personally really believe in that, it's that if you look at one thing in a very monorail way, we won't succeed. And I'll tell you why. Cancer is a very heterogeneous disease. Cancer is not only determined by one mutation. Cancer is complex. And the complexity and the heterogeneity of cancer cannot be mirrored in one single mutation, nor can it mirrored by only mutation alone. We need to understand what happens in the tumor microenvironment. We need to understand what happens in the tumor and around it. If we don't, we never find the right patient for the right drug. So cancer with the complexity of the heterogeneity, and the heterogeneity is not only at baseline, the heterogeneity is temporal. If you treat a patient today and you take biopsies and you take biopsies in 3 months later, you're not detecting the same mutation. That's tumor plasticity. And if we don't factor that in our treatment decisions, we will never be able to make a bigger difference in patient outcome. So this is a 10x problem, as some people would believe that. We cannot break down a 10x problem to a single mutation. That's impossible. And that's a bit of a naive thought. And the same will be the truth when we look into the microbiome. If we believe only the microbiome, the analysis of the microbiome will tell us who is responding, that's not going to happen either. So what we need to understand is the microbiome. We need to understand what's going on in the patient itself, cytokines, proteomics, and all of those things. They need to be factored in, and that need to be brought together. Only the microbiome is it's the next hype. Everyone, it's like immuno-oncology. So and so many hundreds of trials are ongoing because everyone believes immuno-oncology is the cure for cancer. It helps the patient. Is it a cure? Well, we will see. Not likely at the moment. Is the microbiome the answer to everything? Certainly not. So I think we need to be more comprehensive, how we look at data and how we combine the data. And that's kind of what we try to do. And of course, we need also to have, also talked in the morning a little bit, longitudinal studies. The best is, of course, to understand what happens potentially from birth to death, so to say. If something like that would be possible, I think many would love that. But I mean, we need to understand what changes to the microbiome over time. And of course, those data need to be open access, and those data need to be made available in order to analyze it and to drive conclusions out of it. So what we're doing in the SGG and in the IMI world, there's a so-called CrossS2G Initiative. CrossS2G means across diseases. So that means basically oncology looks at it, infectious disease looks at it, metabolic diseases, neurodegenerative diseases looks at it, so diabetes looks at it. That's kind of where we are at the moment, and this is kind of perfectly suited to understand technical differences, technical issues, data standards, data analytics, bioinformatics, etc., etc. Again, what's the normal microbiome? Is the microbiome of a diabetes patient very different from an oncology patient, from an infectious disease, inflammatory bowel disease, God know what? What is the baseline? And of course, what's the baseline based on the lifestyle? What's the difference basically of patients who are, as I said before, smokers, non-smokers, probiotics, prebiotics, whatever it is? That is all data which we need to understand independent of the disease. And that's what we try to tackle in an initiative CrossS2G. The other part is what we're looking into the oncology part. And that's specific, as diabetes has a specific project ongoing to understand incidence. Can they prevent it? Can they predict diabetes and type 2 and others to happen? So what we look into is the same what I discussed before, especially response prediction and or basically as an enrichment strategy for clinical trials and for outcome. And of course, as I said, this goes really also then deeper, that it's not only in oncology, it's also happen in other parts. So the key deliverables, what we hope we can get, is really a kind of a stool data bank, where we really kind of put together all the data we have collected across the SGG, across the diseases, and of course across the indications. And that we can really look into what differs from a patient from disease to disease, preconditions, etc., etc. The other, of course, is also what clusters do we need to define, identify, what clusters do we need to analyze, so to say. There has been arbitrarily a little bit A, B, C. How far do we need to go in order to understand the patient responding or not responding? And of course, what can we do in order to influence the microbiome to have a better response, have a longer response, have basically getting a patient on the long tail of the clinical trials, etc., etc. So that's really enhancement, duration of response, etc., etc. So if we, as an SGG, as an IMI part, and also from an industry perspective, if we can deliver on those three, or at least some of them, I think that already makes a big difference. That brings me to the end, and I thank you for your attention and open up for questions and discussion. There's two on this side.
Okay, now it works. So we have not so much time for questions. So do you have anything that you want to ask? Wolf, please.
Yeah, thank you very much. I think exactly the right question. I was wondering if just by investigating the microbiome, or if we do need to investigate the whole microbiome to get the answers. We saw today an example of the branch-chain amino acid as an indicator for insulin resistance. So I'm wondering if there is not easier accessible matrix like blood where you see the reflection of the microbiome. You see then branch amides up or down, or tryptophan as immune up or down. So that would be my question, that there are pathways, critical pathways, basic pathways that might give you an idea of the composition of the microbiome even.
I'm fully with you. I mean, I think the question is, can metabolics be a surrogate of the microbiome at the end of the day? And I see great potential for that. So I actually think this metabolic research is completely underrepresented in what we're doing in order to understand patients. That comes back to what I said. We need to be a lot more comprehensive. We're looking now, what I say, we're looking at the patients horizontally, meaning we're looking at mutation, 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 or at best PDL1 and so on and so forth. What we need to understand from the patient is vertically. We need to understand the mutation, but the depth of the mutation, and we need to understand the transcriptome, the proteome, the microbiome, and the metabolome. If we bring those together and we can kind of we can put that together, so to say, as an identifier for patient, and even you have the same mutation, you're not having the same transcriptome, you definitely don't have the same proteome, and absolutely for sure not the same metabolome. And if we do it this way, vertically instead of horizontally assessing patients, I actually think we're making a much, much bigger difference.
That's a kind of GWAS approach. You could do it top down or you could do it bottom up.
Right.
Okay, so we have a question from Dusko.
I'm here. Yeah, so I really enjoyed your presentation and was so glad that there is so much congruence between your views and what I tried to expose.
Yeah.
Perhaps because we also work with Laurent Zipogel and had a couple of papers coming out.
Sure.
A question about cohorts. Are you suggesting that IMI develops these cohorts or adapts some of the existing cohorts, or perhaps uses the samples that were collected previously in cohorts, but there is some longitudinal distance and perhaps some outcomes in order to find out this predictive kind of responses that we would all love to have. What of the, three alternatives could have could be most realistic? I think I would know what I would answer, but it would be nice to hear what's your view.
I think we have to have a multi-pronged approach. I don't think there is one way which solves it. I think if we have collected samples already and we have them in the bank, we should then make available and should investigate them. And that's something basically which will help us if we want to understand it. And this is always the critical piece on all of those translational things. If you do it on a genome base, on a proteome base, or on a microbiome, you need clinical data. So without clinical data, I'm not saying it's useless, but it's less useful then you have clinical data. So if we have clinical data associated with samples, that we should use right now. The second thing is basically we should see in which trials it really makes sense to understand the collection of microbiome, and to basically your question, not only that we need to have basically plasma blood collection at the same point in time in order to understand is there a reflection on the metabolome, so to say, as an additional component, as a surrogate, I don't know, but we will see. So that's I think we need to do in a multi-pronged approach and not only one way.
So thank you. Another question, Mark?
So Stefan, you mentioned several issues that are either SGG specific or trans SGG.
Right.
So how would you coordinate these various actions? Because I guess that, for instance, your approach in the SGG oncology will rely actually on the results that would be generated through the trans SGG approach.
Yes, yeah. I mean, this CrossS2G approach basically has been initiated and spearheaded, so to say, by IMI. So the IMI office basically had a workshop in May.
In May,
right?
yes.
Yeah. In May here in Brussels, where we kind of all the SGGs came together with experts and so on and so forth. And the next step is really, and it was consciously put together, this microbiome session today as a learning, as well as basically a reflection where we are. The next step is basically that the SGG has on a cross part to agree what we're going to do and how we're going to do that. That hasn't happened yet, but that's definitely the next step. Yeah.
So the program proposed for SGG oncology will be implemented after?
No, what we do in SGG oncology is independent from the CrossS2G. But what I'm saying is we should answer the same questions independent of the disease in order to drive standards.
So you need to have a cross-feeding mechanism.
Yes.
Between
Yeah,
these various
exactly.
Okay, thank you. Any other question from the public? So you?
We heard earlier on about, from the panel about data access and data sharing. So I was wondering if you could say a little bit more about the plans around making this data available, not only between the SGG but to the wider community as well.
Sure. Yeah. I think in most of the cases, the sensitivity to those data, I think, can be managed. And I'll give you one example. If we have every trial, I mean, unfortunately in one way, but for that one may be actually an advantage, we still run standard of care arm. Yeah? So if you run standard of care arm in those clinical trials, there is no secret. Yeah, because we know the outcome of the patients on the standard of care, because we have seen it 10 times, 20 times, and there's no IP associated to that. So even we just as an industry want to contribute this kind of data to such an initiative, I think that's a great advantage. And if there is sensitivity in certain companies, in certain circumstances, in certain stages of development, those things need to be respected, and that's clear. But this is only to the experimental arm in a clinical trial. This is not to a standard of care arm. Yeah. And chemotherapy, paclitaxel in breast cancer, weekly paclitaxel or whatever you do, I mean, how many trials have been done with that? So we know the results, plus minus 10% or something, and that's not a sensitive piece of information.
Thank you. Any other question? So I have one perhaps to conclude before going and have lunch. So you mentioned this long-term longitudinal study that for studying many parameters across disease, so the microbiome fluctuation and clinical data. So how long these studies should be in your opinion, practically? Because you mentioned before pregnancy and birth, so how do you see it?
I mean, there's twofold to that. One is basically, I would say, kind of a basic understanding how the microbiome changes over the lifespan, so to say. And that's a very interesting question, because that can be indicative of onset of disease. This can be indicative basically of kind of changing in the pathophysiology of certain things. And that's something which is kind of, I would call it more an academic or medical academic question. If that can be dealt with in IMI, I don't know. The other piece is basically, if I have a patient on treatment, I really need to longitudinal follow it. Because I cannot only take a snapshot at baseline and maybe at disease progression again. That's already too late. I need to understand it much earlier in order to predict which patient may or may not do. So there's twofold. One is kind of a patient longitudinal within a disease, and the other one is basically longitudinal, basically to understand changes over the lifespan. So what's the microbiome pre and post puberty? You know, what basically, if a microbiome changes, is that a patient who is going to be developing diabetes in 10 years or in 5? Or is it basically, can I detect colon cancer by looking in the microbiome 5 years ahead of time? And that's kind of what I mean with the twofold answer.
Thank you very much.